Hey everyone, this is Tom Salemi of DeviceTalks. Welcome back to Neuro Innovation Talks. I'm here with our host, John Murray. He's head of global strategic marketing at J&J MedTech Neurovascular. John, this is our second episode, our sophomore episode. How are you feeling?
I'm great today. Thank you, Tom. Good to see you.
Excellent. And this is so. Are you tired of signing autographs and all that goes with being a podcaster? It's a glorious lifestyle, John.
It sure is exhausting. I will say, though, we've made a good impact so far. Lots of interest in the podcast series. So thanks for the partnership here.
Oh, our pleasure. I know I've heard you've had a lot of positive exposure at clinical meetings. It's exactly where we want to be. And we've got a very strong follow-up today. You sat down with Dr. Fernando Vinuela. He's a professor of Emeritus at the University of California in Los Angeles, UCLA, Interventional Neuroradiology. And John, give us a little insights on what we're going to hear today from Dr. Vinuela.
Sure, I'd be happy to do so. As you know, Tom, we were very thoughtful in which physicians we wanted to feature in this podcast series, and those were the people that early on helped to define the field. They are the ones that expanded the boundaries and what was possible in neurovascular, and these are the ones that created the trailblazing innovation. So with that, we certainly have included Dr. Fernando Vinuela in the podcast series. Now, I don't want to say a lot up front, because our conversation covers a huge amount. But I think it's important to highlight that Dr. Vinuela had a significant role in the development of the first detachable coil and doing a lot of early research in taking ideas from the bench into technology, and a lot of this advanced aneurysm treatment. But I also want the listeners to know about Dr. Vinuela's incredible personality and how strongly he's tied to his mentees. And everything for him ties back to those personal relationships. He's got an amazing following of fellows that admire him and follow him. So really impressive. But people don't typically see the human side of the key opinion leader in our space, only their reputation and their image. But Dr. Vinuela's heart and humanity really come out in this podcast, as much as we see in his medical contribution. So with that, I think we can jump right into our discussion.
All right, well, before we begin this episode, I want to bring in our sponsor, Confluent Medical. I'm here with Tom Ditter. He's Senior Director of Product Development at Confluent Medical. Tom, Confluent's made quite a bit of noise in the medical device space this year, specifically surrounding nitinol, which has been just really an exciting topic of the year and of the past couple of years. Before the company was Confluent, it was known as NDC, which stands for Nitinol Devices and Components. Can you give us a quick overview of how Confluent got to where it is today and how your history is connected to NDC and to nitinol?
Yes, before it was called Nitinol Devices and Components, it was actually known as Nitinol Development Corporation. And the philosophy of the founders was to understand the fundamental material science behind nitinol and to use that to design and process components made from nitinol. Now, Confluent continues its legacy today throughout its capabilities. It focuses on the material science of not just nitinol, but also polymers and textiles as well.
That's really interesting. And as a contract manufacturer, your job is to assist OEMs in taking a design from prototype to production. Can you give us some examples of how Confluent's accomplished this recently? Few cases you've worked on. I know you have to probably be sparse on details, but what are some of the things you've been working on?
Confluent is fully integrated and has the capability to take a product from prototyping to development, optimization, and full-scale manufacturing. We recently just worked with the OEM on a novel neurovascular stent. We assisted them in initial design iterations, process development, to full-blown manufacturing.
Tom, Confluent Medical is a contract manufacturer, which means you assist OEMs in taking a design from prototype to production. Can you give us an example of how Confluent has recently accomplished this?
Yeah, yes, as you mentioned, Confluent is fully integrated. We do have the capability to take a product from initial prototyping to full-scale manufacturing. We recently accomplished this with a OEM that was working on a novel neurovascular stent. We worked with them on the initial design iterations, took it to process development, optimization, and finally to full-scale manufacturing.
I thank you for joining us today, Dr. Vinuela. It is an honor to have you as part of this podcast. Now, you're considered a pioneer in the neurovascular field today, but the field literally did not exist when you started your career as a physician. What was it that drove you to commit your energy and resources to addressing the unmet needs in neuro disease?
Well, thank you for asking that question.
Sure.
I think it's very important, John. There was literature in interventional neuroradiology at the time. It was very scanty. There were an article in 1974 by Professor Servinenko from Russia. I remember Charles Drake, an iconic figure in neurosurgery, who immediately published Dr. Servinenko article in General Neurosurgery without even do any change. So Servinenko in 1974, Gerard de Brun in 1976. He had an article also in utilization of detachable balloons, particularly for the treatment of carotid cavernous fistula. There is an article by Charles Kerber on the utilization of NVCA in the treatment of arteriovenous malformation. There was also article from the French colleagues on the utilization of the sternal carotid artery for embolization of dural fistulae, utilizing as a champion a 4 French catheter. And again, there was another article by Jean Jean and Jacques Merlin on utilization of angiography for the evaluation of the spinal cord. So there is a launching pad, a very important one, that in the 1974-76. In 1974, when I doing my residency in radiology, I decided to ask the chairman if I could do a 3rd year of neurosurgery and neuropathology. And the answer was, why? Because I said by heart, I am a neurologist. That was accepted. So my 3rd year, I did 6 months of neuropathology and 6 months of neurosurgery. That I discovered a world unique. In London, Ontario, at the time, there was the only place probably on earth that there was a department of neurosciences. There was a department that conglomerated neurology, neurosurgery, neuropathology, neuroradiology at the same level. At the same time, this department had one every week on Tuesday. The whole town stopped and gathered in one hospital every Tuesday. And the colleagues of that particular hospital deliver different cases to the other ones without knowing what it was. So that was an atmosphere, John. Amazing. There was not, in my many years, 13 years in London, Ontario, I never felt that there was a pyramid. The big leaders were at the same level as anybody else. So there was no intimidation. There was no type of discrimination. So all of a sudden, a little guy coming from Uruguay, I was one of them. I was one of them that opened all the doors. So on top of that, when I did my residency and I was doing neuroradiology, I asked my chairman to send me to France. I wanted to work with Professor Gerard de Brun, who was the Western, for me, is a father of Western intervention. Servinenko is in Russia and Eastern. Gerard is in the Western. So I wanted to go and work with him. And listen to this, only in London. So the chairman said, no, you cannot go. I said, why? Because Charlie Drake just recruited Dr. de Brun to come here, to London, Ontario, Canada, a little town. Gerard de Brun parachuted over there. All right. So that was, incredible. On top of that, because Gerard de Brun was there and I worked with him, I asked the chairman, can you send me to Pittsburgh, where Charles Kerber is doing NBCA? We don't do it in London. Can I go? I went.
Mm-hmm. You got permission for that one, yes.
And I developed an extraordinary relationship with Chuck. He was my professor, then we became friends. And then when I was at UCLA, I was able to retribute him when he needed a lab in San Diego. It was our lab. So he utilized the UCLA lab on that. So that, when I returned, I was bringing the utilization of endovascular technique above the circular Willis for brain AVM. So 2-0, if you want to call it, 2-0. And the third component of that was that when I do it, when I start becoming the first year of a junior neuroradiology at UCLA, Professor Charles Drake, smoking his pipe every week, was coming down with a chair full of cases from all over the world for us to read them. So that was sitting, we were reading it, the diagnostic part, and I was listening to his recommendations. And not unusually, he would say, Fernando, can we help you with that? I say, Charlie, no. How can I do? Do something. Can you imagine the atmosphere? You're talking about a world, number one, extraordinary human being, world recognized for his honesty, asking, can you do something about it?
Yeah, but I wonder, I know what you're going to say when I ask the second part of the question is, was there an aha moment? And it sounds like the way that you described it, there were lots of things that came together. But, what I'm interpreting is, this ability for you to interact with some of the greats at their level. They were open to your feedback and your engagement. Is that how you see it?
I think the way I see it is that there was always time to do something different. There was always time to think about it. There was time to share it. And there was always time that we had an animal lab where to respond it practically in a specific problem. And also we have Gerard de Brun, the big patron, who came to London, Ontario, to a technique that nobody had in the Western world. Nobody had it. And then with his, how can I say it? The relationship was at the beginning relatively strict because Dr. De Brun was a Grand Patron, and in France, the Grand Patron does everything. Does the puncture, put the catheter, So, but I was there all the time with also Alan Fox and working together. And then, during some times, this is another very interesting situation. I hope Gerard de Brun listened to this. The only thing I could do is do the puncture, and then he would do the catheter. But one day, Gerard came and said, Fernando, do you skate? I said, no, I don't have snow in Uruguay. All right, so I think I'm going to go to skate in the weekend, and then I will come. Well, during the skating, he damaged his left hand. So for 2 months, he was unable to work. So in 2 months, I took over.
You were his left hand. Wow, that's a great story.
I took over. And then the relationship, as I said, I never felt any kind of dominance in whatever I think and I was saying when I was doing. I was taking, in that atmosphere, it was extraordinary.
So in those early days, it sounds like those physicians that were mentors for you, that treated you as an equal, were individuals that you relied on. Are there other individuals that you would call on when you had difficult times and challenges? Because I'm sure there were ups and downs in those days where technology was still evolving and still learning. Who did you reach out to? Who were your colleagues?
The same colleagues. One of the things, that we learned from Charles Drake and the rest of the world learned is that when Dr. Drake presented their cases, presented the complications, absolutely every single time. So, that opened a door of honesty and of capacity to share the good, the bad, and the ugly of a specific case and the limitations of them. And from the beginning, in our how can I say, in our path to started doing interventional neuroradiology at the time, there were questions that we have the capacity to answer in the lab. We have an animal lab. So in the animal lab, for example, we developed, for example, in the embolizations of brain AVMs, Chuck Kerber utilized a syllactic balloon for navigations intracranially. The syllactic balloon didn't have the propensity of an elastic balloon or latex balloon. It was very slowly inflation and almost no deflation. So looking into the brain, we didn't want that. We didn't a system that would allow the balloon to open, to inflate and deflate immediately. And the only thing we did, John, is using a regular needles, using the size of a needle, we deliver the capacity of inflation or deflation. It was a big needle, it will be immediate inflation deflation. If it was a middle needle, it will be the same. So based upon that, when we utilize the technology developed before, we noticed that the inflation deflation of the balloons going intracranially were not strong enough. So we went to the lab and utilizing Gerard de Brun's balloon, instead of inflation, we making a inflation deflation balloon. So we slowly and gently, with the support, of course, of the department and the support of also neurosurgery and neurology, with Dr. Henry Barnett. If you're familiar with Professor Henry Barnett, probably the most famous vascular neurology of his time, they were always curious to know how we're doing, what are you doing, what things will be done, etc. That is an environment that I share openly the good, the bad, and the ugly of our cases.
Yeah, I hear that. Just if I could even ask you personally as an individual, how did you manage that pressure in those challenging cases? Clearly, there was fear. How did you manage that as an individual?
I tend to be relatively conservative, if you believe it. So in cases, for example, of brain AVMs, when we started, first of all, Gerard de Brun was the world authority in balloons. So he was still is our master. We learned from him. He taught us how to do it. He taught us to be conservative. So that part of balloons, I was developing a capacity to deal with most of the issues. For example, it's very interesting, I think. When I went for 6 months to work with Charles Kerber, I had a lot of experience in balloons. He didn't. He had experience, of course, on endovascular utilization of balloons. So when I was there, I was able to do balloon cases, doing the cases, and Chuck Kerber allowed me to do the cases. So we share the technology that I have learned from the master.
Mhm.
And so when I returned from Pittsburgh, and I had learned from the master, Chuck Kerber, the possibilities of embolization using his technique, I noticed that in my hands, there were complications that I could not, I had to stop it and do it again. Namely that I remember the last time I presented my 50 cases of embolizations of AVBMs, I talk about my 7 arterial ruptures produced by the characteristic of the balloon. And the reason why I did it was that I didn't want my colleagues have the issues that I had it. I learned it from Professor Charles Drake to share it with it. So those the new latest balloon were much better, far superior, much quicker. That make an event that was very important. So with that element, and I think we were the first one in the world, thanks to Professor Charles Drake, to embolize the first 50 cases of brain AVM embolizations using the technique that had been developed by Charles Kerber. There was an environment that was open up, that everybody knew what was done, everybody knew the results. So everybody witnessed and what the path that we were taking progressively.
I'm going to shift gears a little bit. You talked earlier about your background in clinical neuropathology, neurosurgery. Really, you were seeking to understand the problem as how I interpret it. And then you set up, and I'm fast forwarding, you set up the first of its kind preclinical research lab at UCLA, which you were able to attract researchers from all over the world to brainstorm and collaborate. Can you talk about how that all evolved?
When I was recruited by Professor Hushan Kangarloo, the chairman of UCLA, and I asked him if we were able to have a animal lab research, and he immediately agreed. And we have, first of all, an old angio equipment, and then Dr. Kangarloo brought us a brand new Philips angiographer. So what we did, and then is, as an obligation from us, was to provide that research lab as a training lab for our fellows. First of all, I never like it. I never was confident of a machine as a training for a fellow. I needed blood, vessels to rupture, how to control. So every single of our fellow have a training in our lab almost twice a month, in which there was the capacity to navigate the intravascular until the skull base, for example, of a dog, which is 2.5 millimeters, or the optic artery of the ophthalmic artery of the swine, which is 1.5 millimeters. In artery, very spastic. In carotid, very smaller. So the capacity of navigations for that was paramount to us. Paramount and obligatory. So from the beginning, that was what the animal lab gave us. And thank you to Professor Kangaloo. And then when Guido came and did all his work in the animal lab and brought the CDC, when we brought Microsoft, a small company, to develop Onyx, which was the first liquid embolication that was not adhesive. Number two, Pierre Gobin, another our fellow from France, used our lab to develop the first ever technology to withdraw a clot. So 3-0, that lab became, I would say, well known.
Mm-hmm.
And that lab was the origin, John, that we had so many brilliant fellows from all over the world. Some of them came from a week, some of them from a month,
Mm-hmm.
some of them from a year, some of them for 7 years. That was a living world. And economically, we had support from our chairman. We had support from NIH grants. We had a few. And support also for money delivered from the CDC coil that went to university, and we're getting a percentage of that.
What do you think drew these fellows? What was it about UCLA that encouraged these brilliant minds to bring this innovation to UCLA specifically? What was the draw? Was it the unique setup in the lab? Was it something that was one of a kind? Is there something else?
Well, I think we have an open, I tell you, our division received the visit of 447 colleagues. 447. No, I didn't train, first of all. But every one of them, our regulation was, whatever we had, whatever he has. Whatever I come to visit, whatever we do, he sees. Whatever we do in the clinic, in the lab, he does it. And if there is anything interesting in mind that he or she like to deliver, we will discuss it. So there was no barriers.
Mm-hmm.
They was just coming totally free, and they deliver whatever they wanted, and they returned always with something. We tried that every single our fellow coming for at least for a year, when they left, at least they have to have 2, mainly 3 manuscript in good journals. That was our responsibility when they come. So that was a good attraction, I think.
So a theme that I've noticed about and referencing the pioneers in stroke innovation was that all of those leaders were known for thinking outside of the box and breaking boundaries, and you're certainly no exception. And one of the areas that you took some risk in was training other specialties, neurologists, neurosurgeons, of course, along with neuroradiologists. Can you talk about why you felt this was necessary and how you survived that?
Excellent question. Well, first of all, in London, Ontario, I worked from 1990 to 1986. And in those years, I trained 4 neurointerventional fellows. Jacques Dion, radiologist, Pedro Lilik, pediatric neurosurgeon, Teresa Lin, Pedro Lilik from Argentina, Teresa Lin from Uruguay, neurologist. And so you can see that even when it was a tiny thing, I was already thinking that there is no boundaries in a specialty. The one who does it the best is the one who should do it. So when I went to UCLA, after a year, I established a 2.5-year program, John. Was the only institution that had a 2.5 years program.
Mhm.
Why? Because we needed a number of interventional for a country. And interventional were minuscule little stand in relation to neurosurgery and to neurologists. So this is what we did. We did a training program that included 4 rotations, 6 months neuroradiology, 6 months neurosurgery, vascular neurosurgery, 6 months stroke neurologist, and 1 year of interventional technology. So depending on where they're coming from, a radiology didn't do neuroradiology, but did the other 3. A neurosurgeon didn't do vascular, but the other 3, and vice versa. So at the end of those years, I have the tranquility, and I was able to provide support to those fellows who have gone through those treatments and who had the right weapons to make a decision. So you understand that I had a lot of criticism for my peer from diagnosis that were saying, why are you teaching somebody to read a scan on MRI? They will remove our cases. I say, are you nuts? They have 7 years of before reaching us, have 6 and 7 years of very intense training, and they're going to do that? They're going to sit in front of MRI to read a question? No way. But listen to that as well, because so on reviewing, and then I want to show off a little bit, if you want to call it, on reviewing which are the presidents of SNIS, okay, I was one, but Gary Dadweiler, neuroradiologist, Jacques Dion, neuroradiologist, Mary Jansen, neuroradiologist, Michael Alexander, neurosurgeon, William Mack, neurosurgeon, and Dilek Yabagal, the first president of SVIN. So there you
Well
go.
done. Well done. And thank you for explaining that. I interpret that as you well-trained those individuals, and they came to you very experienced, and your philosophy around the best and most talented should be doing the procedure. It's logical.
I will take a quick break from this conversation to bring back our sponsor, Confluent Medical. I'm here with Tom Ditter. He is Senior Director of Product Development at Confluent Medical. Tom, nitinol is growing in popularity. We're seeing more and more OEMs are turning to it. This alloy has shape memory. It has super elastic properties. Can you explain how Confluent sets itself apart from other nitinol suppliers?
Well, Confluent has over 30 years of experience in the design and processing of nitinol. But beyond that expertise, Confluent identified a nitinol supply chain gap. They invested over $100 million across the supply chain. Most recently, they just made an investment of over $50 million in additional melt and conversion capacity, becoming the fulfillment partner for one of the largest melt suppliers in the industry.
So, Tom, we're hearing so much about nitinol's super elastic and shape memory properties. It makes it an ideal alloy, specifically within the neurovascular space. Can you speak to how Confluent's expertise is able to meet the growing demands for OEMs in the neurovascular market?
Yes, for neurovascular applications, durability is a major consideration, and Confluent Medical has invested heavily in the development and processing of high purity alloys for increased durability and fatigue resistance. Our material SE508 Eli is one of the highest purity materials available and ideal for high cycle fatigue applications, as found in neurovascular devices. Additionally, Confluent specializes in high precision polymer tubing, such as film cast PTFE. This makes us unique in the neurovascular space in that we can offer PTFE with thinner walls and larger diameters, making it ideal for large diameter aspiration catheters.
So, Tom, that's super interesting about the neurovascular space. So, what does the future look like for nitinol in the neurovascular device market?
The demand for minimally invasive procedures is growing, so we can expect to see nitinol as a key alloy for these devices. Future applications such as neurovascular devices will need to have a smaller profile with an emphasis on durability. Additionally, high purity nitinol is needed to address high fatigue applications to prevent fracture and improve longevity for patients.
Well, this has been really entertaining and insightful, Tom. Thanks for joining us on the podcast.
Thank you, Tom. Glad to be with you.
And thanks to Confluent Medical for sponsoring this episode. If you'd like to find out more about Confluent Medical, go to confluentmedical.com or, interestingly enough, nitinol.com.
So another example, if I can, of thinking outside the box, and this time from a technology standpoint, was your role in the development of the GDC coils. And maybe I might even ask you to describe for our audience briefly what GDC stands for.
With the helmet detachable coil. Well, this is the reason, that our relationship with Guido, I knew Guido from London, Ontario. Guido went to visit Gerard de Bruyne in London and spent there again 10 days and returned to Rome. So, and we continue a friendly relationship for a while. Not for a while, until today, but what I'm saying at the beginning. So when I was recruited by Professor Hushan Kangaroo, and I asked Professor Kangaroo if he could help us to bring Guido for 1 year to work in our lab because Guido didn't have the possibilities of doing it in Rome. Professor Kangaroo immediately agreed. So I sent the letter. I received a letter from Guido in 1987, and I sent a positive response in 1999. So he came on the 1st of January of 1999 to UCLA. So in UCLA, we had a very good laboratory and led by John Roberts, an experienced technical expert, extraordinary personality. And since the arrival of Guido immediately started to work in the research animal lab, and who also had Pedro Lilik as our fellow technically related to the lab as well. I tell you, it was a lot of fun, it was a lot of work, and it was a lot of cigar smoking. Those three guys smoked. So in this manner, the that was the beginning. So Guido was very comfortable there at the time there, and whatever he needed was provided. Mid-year in '89, I called Eric Engelson, who was the leader of Target Therapeutics, a little company that once came to show me a little catheter to put it in the liver, and I say, no, I want to put that catheter in the brain. So and then I told Eric, we'd love to share with you Guido's ideas. Would you be so kind to help us? And can we start this called the Project X? The Project X. So a week later, Ivan Sepeska parachuted here with us, and then there was a team, Guido and Ivan, working with Pedro and all in that environment, starting working in the development of the GDC. We had meetings every 10, 15 days, talking about what what the development, the yes and the no, and how to make the differences. And all of a sudden, that became on its own, working on its own in its most extraordinary environment. But it was hard work, a lot of fun, and everybody was sharing everything. So when that came up, and I had the honor to do the clinical part when the CDC was utilized clinically, I was responsible for the selection of the different centers that were going to be involved at the beginning of a technique, because the important was safety, first of all, because you can understand that we have an audience who was really interested in our preliminary results.
Fascinating. So there was also a lot of stress there as well. Here's a photo of you and Dr. Guglielmi in 1991, and then you 20 years later. I think you shared this photo with me in the past. What comes to mind for you when you see these images? And then any stories that you can tell or emotions that surface between then and that 20-year photo?
Look at the pictures. All right. So what do you see in the pictures? There, you don't see cortex of the brain. You see limbic system of the brain. The limbic system is the part of the brain that makes you happy, that makes you cry, that makes you sad. This is it. So the pictures speak by themselves. I say that, we are in close contact, and we still share, our lives. We have been in contact. He invited me to go to Rome, and when he comes, we have this relationship. That relationship is unbreakable. That relationship, I will say, withstand time and distance, because this is it. It's just whatever happens is a foundation of what we have today.
It's fantastic. So let's shift to mentorship for a moment. You're talking about relationships here. Your mentees that I've talked to have shared that they see you as a friend and a partner, similar to the way that you describe the way that you interacted with your mentors in the early days. You've been a motivator to many of these and lifted them up when there's challenges, and specifically around publishing, you've put your fellows and your partners forward. During your career, I think you've trained 449 physicians, and I know that you consider all of them friends. Can you talk about the importance of those relationships that you've built?
The honor of that our division developed to receive fellows, young fellows, super capable, super brilliant, that they wanted an opportunity to develop their careers. That was our concept first. What do we do for them? If they will stay for more than 6 months, we have the responsibility, as I said, that going back home, they will have at least 2 or maybe 3 papers in the literature. But if we stay longer, the responsibility was start developing their professional careers in ours, thinking in their country. And then we have examples, for example, many examples of young physicians who spend with us 4 years, 5 years, 6 years, 9 years. And then I can give you names, Pierre Gobin, he spent for years, now is a leader. And Yuichi Murayama has been a year. He was the youngest chairman of neurosurgery in Japan. And he was extraordinary. He is still extraordinary. We had the fortune of having him for such a long time. And we have Ishiro Yuki, 5 years, now working in the universities, etc. So, and then why first? Because the concept was the guy who brought the idea. No, he didn't bring the idea. Sometimes they are coming from us. The guy who worked hard, write the paper, and does the job, and the old guy is the last one as a mentor. A mentor. It's a time in which, so that's the concept, that's the rule. That rule, it is persist. And then you see that we believe that it has been working.
Now, we mentioned 449 physicians. They really all consider you a friend. How do you keep up? They tell me that every time I text Dr. Vinuela, he gets right back to me. He's always available for me. That's a heavy lift there. How do you manage that?
Oh, John, it's not a heavy listage.
That's a lot of friends.
Yes, of course. I mean, it's a and as well, John. The word opportunity is that when they were finishing the training, and they were going back home, and they have patients, difficult patients, let's say politicians or whatever, and one of us we're going to help. One of us we want to help, and it means it could be Greece, could be Argentina, could be anywhere, Spain, because that was part of our. We never signed a contract, but it was part of the protection in the early phases of them. That quality and security is one. I think we have the responsibility to help.
We've heard a theme emerge in this discussion about you not being afraid of challenges and having an insatiable desire to learn new things as you've represented in your clinical background. So when you left UCLA, you took on another challenge by helping patients in Brazil by advising and educating on stroke systems of care there. Not to mention learning Portuguese. Can you talk about what you've been doing recently in Brazil and some of the initiatives that you've been involved with there that we may not know about?
I'd be delighted, John. And this is an example that you never close the door to life, and whenever the train stop, you better get in if you want. Don't hesitate. John, this is very interesting because I already had plans to retire from UCLA in June 2013, and Gary was ready to take over. In March 2013, we received a patient from Brazil, a lady from Brazil who had a unruptured aneurysm and the anterior circulation, and they want us to treat her. So the lady came on a Thursday, we treated her on a Friday, and she returned on a Monday to Rio. 15 days later, my wife and I received a letter from Brazil from the husband of our patient inviting us to visit her beach house in Brazil. So my wife and I went, and then the first shocking thing was that there was a helicopter waiting for us in Rio to take him directly to the house. In the next morning in breakfast, I realized through people there that the husband of the patient was the co-founder and of the largest Brazilian healthcare service covering 5.6 billion people in 33 hospitals. So,
wow.
And the invited people were 6 hospital directors
This was a surprise to you. Wow.
So there you go. So I spent a beautiful weekend, a few days, and when I returned, one day it was something like this. And I wake up at 3 a.m. and say, okay, I know what I want now. So, and he wanted me to develop a program of interventional, neurointerventional therapy in his in his company. But on the other hand, I tell him, no, I like to develop a interdisciplinary stroke program with inclusion of interventional. And we call it PAVN, Program Interdisciplinary Program of a Stroke and Neurointerventional. That, the reason why I thought I could do it is there's another issue here which is related. You know, Professor Hushel Kangulu, when I was working with him, promised that he would help me to open the doors to the South, to Latin America. And thanks to him, in September 1989, we developed CILAN, the Ibero-Latin American Society of Interventional in Mexico. 1989, thanks to Hushel Kangulu, we could open the doors to Latin America in a society that Spanish and Portuguese were the ones because Spain, Portugal, and Latin America was part of this society. Today, this society has 34 years, and it's open up interventional, and it opened up over there. So the program, instead of only being limited to interventional, was acute stroke. And the reason I thought I could do it is because during the years of developing CILAN, I was able to meet, without exaggeration, John, hundreds of young people from Mexico to Tierra del Fuego. Each country we visit every year until each country had the Society of Interventional. And I was able to know in Brazil who were the physicians that probably will be in keeping with organizing a interdisciplinary organization that included the 5 members of a stroke meeting, a strong team throughout the country. So we started first having a hub in Rio and a hub in Sao Paulo. And then from there, we worked from 2014, 2017, working day and night to develop this kind of a stroke society, a stroke organization, which still now work. I finished in January 2017.
Excellent.
So I tell you, it was an extraordinary experience. I had to get Portuguese as my second language, and not my second language, but to learn Portuguese. And I still have all my relationship with a myriad of Brazilian friends.
That is no surprise to me at all. So what listeners here may not know about you is that you sign at least some of your emails fraternal hugs, even to me. I think it says a lot about you as a person. The idea that we're all in this together, regardless of where you come from and what your role is. But to me, it also reflects how you've brought different disciplines together, You've certainly prioritized your personal relationships in your work and life. And I think you've instilled in your colleagues that it's not just the work of one person. We all do this together. This has been very clear. That we're all coming together to solve something bigger. But this philosophy for me reflects a huge amount of humanity and very little ego. Where and how did this way of thinking originate for you? It could go two directions, right? It could be, I am the smartest guy in the room and I've been able to solve these problems, or it could be, we're in this together. Where does that come from, Dr. Vinuela, for you?
John, this is something that very touched to me. And first of all, I was born in a tiny city, 30,000 people. And I was one of 14 children, one of 14, that my aunt and my mother grew up together. We were 14 children.
Wow.
Now, you understand the sharing in 14 children. Because some of those children were from my mother's brothers, my uncles, who were working in the fields, and when the children were in the school age, they were bringing them to my grandfather's house, and then my aunt and my mother would take care of them until finish secondary school. So there you go.
Excellent.
I was in the middle. So okay, that's one. Number two, my little country, Uruguay, has been a leader in equality. If you look at the laws, if you look at the way behaviors, social behavior, the community behaviors, our education, primary, secondary, university, was based upon no inequalities, no discriminations. I never ever heard that I want. That's number two. And number three was basketball. Because I played basketball for many years. I play in my hometown. I play when I'm in medicine. I play when I was studying medicine. I was a private institution called Aguada. Was kind of a high. So I play at different levels since I was 15 until I was 30. So in a basketball, what did you learn? You learn to share the good, the bad, and the ugly. So, I had in mind all my time, and also, that I brought that. I brought that up. I was in Canada, and I was with a concept that there was no inequality whatsoever, and that's a reason.
That's excellent. Those are good explanations. Last major question for me. In addition to, of course, hard work and focus on the patients, which you've represented here, what advice do you have for physicians and industry entering the neurovascular space today?
I have the luckiest man in this world. My wife and I have a 21 years old girl, and she's now at Johns Hopkins working, and she loves cognitive neurosciences. And I ask myself, what can I tell my daughter? How can I call my doctor? How can I help my daughter to face her immediate future in a world that is changing every week? So that question, if I have to answer it, and you push a gun on me and say, Fernando, tell something about it. So I say, well, yes, I can use which I learned as a mentor. I can use how to be a friend for my mentor. help my mentor very carefully, take care of him, make the best for him or her, and be prepared to answer any kind of question personally or whatever, and share my own weaknesses in life, etc. So this is what I can say in a relationship between a mentor and a mentee, or a mentor and that. And then the rest is so difficult to me. And when I'm talking of mentor, I'm talking as a patient as well. There's the concept of using something that the IA doesn't have, which is limbic system. We're humans.
Mm-hmm.
The AI still is not human.
Yeah.
So if we still follow the concept of those concepts, I think it's something that we can still be useful. And but it's a question that I will never be able to deliver to my daughter, because I don't think I will have the capacity to look at the immediate future and trying to learn where are we going.
Yeah, no, I understand. Thank you for trying to answer. I have a next session, and then we'll wrap. It's lightning round questions. This will be rapid fire, just quick answers. If you could invite any 3 historical public figures, dead or alive, to a dinner party, who would they be?
Nelson Mandela would be one. Jose Artigas, which is a George Washington of my country. And Steve Jobs, that will be the third one.
Excellent. If you had only one book to read for the rest of your life, which would it be?
I tell you what, John, I have the book nearby. It's a Brazilian book. When I was still learning Portuguese, I chose a book called, let me see if I remember that, Brazil, uma biografia, a biograph of Brazil, written by 2 ladies, Lilia Schwartz and Eloisa Starling. It was the most extraordinary book in history.
Would you say you're more creative in the morning or the afternoon?
Early morning. I'm an early bird.
And what is one talent that you wish you had that you do not?
Music.
And your favorite food?
Seafood.
And if you had not gone into medicine, what would you be doing today?
Oh, you will love that one. Director of a symphonic orchestra.
Connected to music. Wonderful.
So, but that I will make today to you. When I was in my hometown, when I was 13 years of age, there was no TV, but there was a radio. And there was a special radio in Uruguay that had classic music. And I would listen to the music. I was pretending I was I was directing it.
I can see you as an orchestra lead. Yes. The maestro.
If you want to call it that.
So before we conclude, is there anything else, Dr. Vinuela, that you would like to talk about before we wrap up the session?
John, it has been an extraordinary time to me, because, as I said before, I never stopped to smell the roses, and I never stopped to figure out and put in my brain how, when, and where the situation happened. I live in my country in July 1st, 1974, and it was a road that I never went back to watch until you told me. And so I really thank you dearly for this opportunity.
I thank you for your openness, transparency, and willing to address any question, and just your significant impact on patients' lives throughout your career. It's immeasurable. I look at this all as a privilege to be here with you today, and I mean that. So I feel that I've been added to your 449 trainees. So I'm now 450 because I did visit your lab in the mid-90s when you were very gracious and invited me in to observe cases. I'll never forget that day many, many years ago. So thank you for your leadership, for being a pioneer, and for leading the way for many other physicians to help patients' lives.
Thank you, John.
Thank you.
Well, that is a wrap. Thanks so much for joining us on this episode of the Neuro Innovation Talks podcast. Thanks, of course, to our friends at Johnson Johnson MedTech Neurovascular. It's been great to work with them, in particular with John Murray, who's spearheaded this effort and really helped us tell some important neurovascular stories. If you don't want to miss a future episode of Neuro Innovation Talks or our other great Device Talks podcast, make sure you subscribe to the Device Talks podcast network. We'll have future podcast episodes sent directly to you. You can also follow us on YouTube. We'll have our audio and video portions or versions up there. So lots of great ways to keep informed and to gain great medtech insights. Finally, make sure you share this podcast episode on LinkedIn and other social media channels. And when you do, connect with me, Tom Salemi, Editorial Director at DeviceTalks, and our great Managing Editor, Kayleen Brown. We'd love to be part of your future MedTech conversations. That's it, folks. Thanks again for joining us on this episode of the Neuro Innovation Talks podcast.